Today we're going to talk about double Punnett squares. And double Punnett squares is when you're trying to find the frequency of having two traits at the same time. So for an example, we're going to do hair color. And so if you have a dominant, you have straight hair, recessive curly, uppercase B, dominant, brown, recessive blonde. And this is an oversimplification, but what we're going to do is we're going to do two people who have straight brown hair but are heterozygous for both traits. So their genotypes will read big A, big B, little b. Now the hardest thing about a double Punnett is setting up the boxes because you have to do all the different allele combinations. So the easiest way to do it is to actually underline it out. So for the first box, for the first parent, it's going to be the first A with the first B. So big A, big B. For the second box, it's going to be the first A with the second B. Big A, little b. The third box is going to contain the second allele, the little a, with the big B. And the fourth box will be the little a with the little b. Okay, now luckily because I did two parents who are the same, it's going to be these same four down the other side. So big A, big B, big A, little b, little a, big B, little a, little b. Now you have to fill in all 16 boxes doing the exact same thing you did with a simple Punnet. You were just bringing it down and across, and each box is going to contain 4 letters, just like it does up here for the genotype. So for the first box, it's going to be big A, big B, big B, because I simply brought these two down and these two across. You always want to put the capital letter first, and you always want to keep the two alleles together. So this should always read 2 A's and then 2 B's, not A, B, A, B. So for the second box, it's going to be a big A, but now it's going to be a big B and a little b. Again, I made sure to put the big B first, the recessive allele second, and I put the A's together and the B's together. And so you're just going to keep filling out your Punnett square. It's going to take you a little bit of time, but eventually you will be able to fill it out and have all 16 boxes. Now the hard part's going to be when you have to count what you get. So when you start to count, you obviously want to have some kind of table over here that gives you an indication so that you can keep track of what each phenotype is going to be. So for this case, big A is going to be straight hair, and big B is going to be brown hair. So this is going to be homozygous dominant for both traits with straight brown hair. And so as you go along, to make sure that you count all 16 boxes, I would cross them out as I go to make sure at the end you are going to end up with 16 different. Now that may not necessarily be 16 different. For example, if I were to continue on, I would figure out that you're actually going to have 4 out of 16 be heterozygous for both traits. So 1, 2, 3, 4. So 4 out of 16 are going to be big A, big B, little b, which means that they're going to have straight brown hair, but they're going to be heterozygous for both traits. And so if you keep following this process, you'll have no problem understanding how to do a double Punnett square. Good luck.